Welcome to Genomics Daily Brief, your shortcut to understanding today's most fascinating genomics news.
Okay.
We've got 3 preprints from ArXiv, all published today, February 13th, 2025.
All right.
Get ready to explore the origins of satellite DNA,
Okay.
the potential of AI for predicting cellular responses,
Interesting.
and the innovative world of variational causal inference.
Sounds like a fascinating deep dive.
Let's jump right in. We've all heard about satellite DNA, those large repetitive sequences in our genome whose function remains largely a mystery.
Right, a significant portion of our DNA, but still an enigma.
A new study on jewel wasps,
Oh, wow.
Mazonia vitripennis,
Okay.
suggests a surprising origin for alpha satellite DNA,
Really?
mitochondria.
Mitochondria. That's unexpected to think those tiny powerhouses of the cell could hold the key to understanding these enigmatic sequences.
Researchers found alpha satellite-like sequences within the wasp's mitochondria organized in palindromic arrays. They actually increase the mitochondrial genome size significantly.
Interesting. So how did they make the leap from mitochondria to the nucleus?
They discovered nuclear copies of these sequences nestled near NMTs, which are essentially bits of mitochondrial DNA that have migrated to the nucleus.
So the proximity to MTs suggests a transfer from the mitochondria to the nucleus. It really challenges our previous assumptions about satellite DNA. This finding suggests a much more dynamic and potentially crucial role, perhaps even in chromosome stability.
It's the first time a mitochondrial origin for satellite DNA has been proposed.
Wow, groundbreaking.
And it seems these sequences didn't just sit there. They evolved rapidly in the mitochondria, likely due to GC-biased gene conversion.
GC-biased gene conversion. So the DNA copy machinery in the mitochondria seems to prefer using the building blocks G and C, more often leading to changes in the DNA sequence over time.
Exactly. And once they hopped over to the nucleus, the nuclear copies underwent significant expansion, forming complex higher-order structures.
So they were evolving both in the mitochondria and then again in the nucleus. Fascinating.
And the researchers found similar patterns in another wasp genus, Macidiphyuax. This hints that this mitochondrial origin phenomenon could be much more widespread.
It makes you wonder what other genomic elements might have unexpected origins. Now let's switch gears to a powerful tool in genomics: variational causal inference.
It's a statistical method that allows us to predict outcomes under different conditions. Essentially, lets us explore what-if scenarios.
Precisely. What if we could predict how a cell would respond to a drug without actually exposing it? Variational causal inference offers that possibility.
We have 2 papers today that focus on this approach, both highlighting its application in predicting cellular responses. The first paper tackles a significant challenge: predicting outcomes when you have high-dimensional data like gene expression, but limited information about other influencing factors.
The classic problem of having tons of data but lacking crucial context.
This method cleverly uses 2 sources of information to construct a counterfactual outcome.
Okay.
First, it looks at the individual's observed outcome, the actual data we have.
So learning from what actually happened.
Right. The second source is the response distribution of similar individuals who received the treatment we're interested in. It's like looking at a group of comparable individuals to see how they reacted.
Combining individual and group-level data to paint a more complete picture.
The second paper, predicting cellular responses with variational causal inference and refined relational information, takes this concept and applies it to a very specific problem, predicting a cell's gene expression under counterfactual perturbations.
So predicting how a cell would react to a condition it hasn't actually experienced.
Exactly. To achieve this, they incorporate gene regulatory networks, or GRNs. These GRNs map out the relationships between genes, showing how they regulate each other.
Imagine a complex network of traffic lights controlling the flow of vehicles through a city. That's analogous to a GRN, where genes are the destinations and regulatory elements are the traffic signals controlling their expression.
The researchers didn't just use pre-existing GRNs, they actually refined them during the model's training process, leading to more accurate predictions and new insights into gene relationships.
So the model isn't static, it's learning and evolving its understanding of these intricate networks. It's like the model is becoming a better biologist as it analyzes the data.
They also developed a robust estimator for something called marginal perturbation effect, which quantifies the overall impact of a perturbation on gene expression.
So not only can they predict if a gene's expression will change, but they can also measure the magnitude of that change. That's impressive.
Right. This is a novel aspect that previous work hadn't addressed.
Very cool.
These 2 papers showcase the power of computational approaches for unraveling complex biological processes.
And we've only just scratched the surface of their potential applications. Think personalized medicine, drug discovery, and so much more.
Speaking of uncovering hidden secrets, that study on the mitochondrial origin of satellite DNA really makes you think.
It's a stark reminder of how much we still don't know about the genome and how many more unexpected discoveries await us. what other genomic elements might have surprising origins.
And what about the function of these alpha satellite-like sequences? We know they're expanding and evolving, but what role do they play in the cell?
Perhaps they contribute to chromosome stability, gene regulation, or some function we haven't even imagined yet. It's a tantalizing mystery that researchers will be eager to unravel.
It seems with each new study, we uncover more questions than answers.
That's the beauty of science. The more we learn, the more we realize how much more there is to discover. It's incredible to think that these tiny mitochondrial sequences might have such a big impact on our genome.
Right. The implications are huge. Let's dig a bit deeper into this study.
Okay, can you remind me what exactly is alpha satellite DNA again?
It's a specific type of satellite DNA found near the centromeres of chromosomes.
Okay, those crucial regions that help separate chromosomes during cell division?
Exactly, essential for cell division.
Right.
These alpha satellite repeats are basically long stretches of repeating DNA sequences,
And their role?
still not fully understood.
So what did this study uncover?
Well, the researchers were examining the genome of the jewel wasp Masonia vitripennis,
Okay.
and they found something remarkable: over 1,500 alpha satellite-like repeat units scattered throughout the wasp's nuclear genome.
That's a lot of repeats. What's so special about them?
Well, 39 of these repeat units were organized into two palindromic arrays within the wasp mitochondria.
Hold on. You're saying they found alpha satellite-like sequences in the mitochondria? That's pretty unusual, isn't it?
Very unusual. And these mitochondrial arrays weren't small either. They actually increased the mitochondrial genome size by a substantial 50%.
Wow, that's a big change. So how did these sequences get into the mitochondria in the first place?
The researchers think these sequences actually originated from the nucleus, specifically from 10 independent insertions of mitochondrial DNA into the nuclear genome.
So it's like a reverse migration. We usually think of nuclear DNA influencing the mitochondria, not the other way around.
Exactly, and they found evidence to support this. They discovered that the nuclear copies of these alpha satellite-like sequences were located near NTs,
Which are?
those segments of mitochondrial DNA that migrated to the nucleus.
Right, those bits of mitochondrial DNA that took a trip in the nucleus. So basically, the location of these sequences near NUMTs supports the idea of a mitochondrial origin.
Exactly, and it doesn't stop there. The nuclear copies underwent a huge expansion, increasing their copy number dramatically.
So this wasn't just a one-time event. It's like an ongoing process of expansion and evolution.
Exactly. One specific array called Saddle L4b showed a remarkable expansion, going from just 1215 copies to over 400.
Wow, that's incredible. What could have caused such a massive expansion?
Well, the researchers noticed a fascinating trend in the evolution of these sequences within the mitochondria. They found that these sequences evolved rapidly with a significant increase in GC content,
GC content meaning?
the proportion of guanine and cytosine bases in the DNA sequence.
Right, so the mitochondrial environment seems to have favored sequences with higher GC content.
It seems so. This increase is likely due to a process called DC-biased gene conversion. It basically favors the incorporation of guanine and cytosine bases during DNA replication.
Interesting. And what happened once these sequences moved to the nucleus?
They continued to evolve, forming complex higher-order structures through duplications and rearrangements.
It's like they were building upon themselves, creating increasingly intricate patterns within the nuclear genome.
And to add to the intrigue, the researchers observed similar patterns of repeat insertions into the nuclear genome in another wasp genus Macidiferax.
So this isn't just a quirk of a single species of wasp?
Right. It suggests that this mitochondrial origin of alpha satellite-like sequences could be much more common in insects.
This study really changes our understanding of satellite DNA. It makes us rethink their origins and their evolution.
It definitely challenges our assumptions and reminds us that there's still so much we don't know about the genome and how much more we have yet to discover.
Absolutely. It highlights the power of curiosity and questioning what we think we know.
For sure. This study raises many new questions for future research.
Like what, for instance?
Well, if alpha satellite DNA can come from mitochondria, what other genomic elements might have surprising origins?
It's a fascinating question. What other unexpected origins might be hidden within our genomes?
And what about the function of these alpha satellite-like sequences? We know they're expanding and evolving, but what are they actually doing in the cell?
Do they contribute to chromosome stability, gene regulation, or maybe they have some function we haven't even thought of yet? It's an exciting mystery for researchers to unravel.
It really is. With every study, it seems like we uncover more questions than answers.
That's what makes genomics so captivating. There's always more to explore and discover.
And with all the advancements in sequencing technology and computational analysis, we're in a great position to make even more groundbreaking discoveries.
It's a thrilling time to be involved in genomics. We're uncovering the secrets of life one study at a time.
And who would have thought it would all start with a tiny wasp and its unique collection of repeating DNA sequences.
It's amazing what we can learn from even the smallest creatures.
Well, that brings us to the last story for today.
I'm ready.
Remember that powerful tool we talked about earlier, variational causal inference?
Yeah.
We have 2 more papers that dive into its application in predicting cellular responses.
Sounds interesting. Tell me more.
The first paper, titled Variational Causal Inference, tackles a big challenge in genomics and other fields that work with high-dimensional data.
What kind of challenge?
The challenge of predicting outcomes when you have limited information about the factors influencing those outcomes. They call these factors covariates.
So, for example, trying to predict a gene's expression level but not knowing much about the environment or lifestyle factors that could be playing a role. It's like solving a puzzle with some pieces missing.
Exactly, and that's where variational causal inference comes in. It uses a clever approach by combining 2 sources of information. The first is the individual's observed outcome, the actual data we have for that individual.
So in our example, that would be the actual gene expression level we measured, essentially learning from what really happened.
Right. The second source of information is the response distribution of similar individuals who received the treatment or condition we're interested in.
So we're looking at a group of comparable individuals to see how they responded to the same treatment.
Precisely, by combining individual and group-level information, this method can make more accurate predictions, even with limited covariates.
It's like filling in those missing puzzle pieces by seeing how others have solved similar puzzles.
Now, the second paper, Predicting Cellular Responses with Variational Causal Inference and Refined Relational Information, takes this concept and applies it to a specific problem predicting a cell's gene expression under counterfactual perturbations,
Counterfactual meaning?
conditions that the cell hasn't actually experienced.
Oh, I see. So like trying to figure out how a cell would respond to a certain drug without actually giving it the drug.
Exactly. This method lets us explore those what-if scenarios without actually doing the experiment.
That's pretty powerful. How do they do that?
They do it by incorporating something called gene regulatory networks, or GRNs, into their model.
GRMs, what are those?
They map out the complex web of relationships between genes, showing how they regulate each other.
So like a map of how genes interact and control each other's expression.
Exactly. Think of a busy city with a network of roads and traffic signals. The genes are like destinations, and the regulatory elements are the traffic signals controlling their expression.
Interesting analogy. So how do these GRNs help them make better predictions?
They provide really important biological context. By understanding how genes interact, the model can make more informed predictions about how a cell's gene expression would change under different conditions.
It's like having an expert guide to help us navigate the complex world of gene regulation.
And get this, the researchers didn't just use existing GRNs, they refined them during the model's training process.
So the model is constantly learning and improving its understanding of gene relationships. It's like the model is becoming a better biologist as it analyzes the data.
Exactly. This refinement led to more accurate predictions and even gave new insights into how genes interact.
It's amazing how the model can not only make predictions, but also improve our understanding of biological processes.
And they didn't stop there. They also developed a robust estimator for something called marginal perturbation effect.
Marginal perturbation effect, what is that exactly?
It's a measure of the overall impact of a perturbation on gene expression.
So they're not just predicting if a gene's expression will go up or down, but they can also tell us how much it will change.
Exactly. It gives us a much more detailed understanding of how perturbations affect cellular systems.
That's really impressive. These 2 papers showcase the incredible power of computational genomics. They show how these approaches can help us unravel complex biological processes and make predictions that were impossible before.
It really feels like we're entering a new era in biology where computational tools are essential for understanding the complexity of life.
And it all goes back to the incredible complexity and beauty of the genome. There's still so much we don't know and so much to discover.
Absolutely. These studies remind us that with each new discovery, we uncover even more mysteries waiting to be solved.
It's a never-ending journey of exploration and discovery.
We've talked about uncovering hidden secrets and powerful tools. Let's circle back to those 2 papers focusing on variational causal inference.
Yeah, let's dive into those.
The first paper, simply titled Variational Causal Inference, tackles a really common challenge
Okay.
in genomics and other fields dealing with high-dimensional data.
What's that?
Predicting outcomes when we have limited information about all the factors influencing those outcomes.
I see.
They call these factors covariates.
So like trying to predict a gene's expression level, but not having all the information about a person's environment or lifestyle?
Right. It's like solving a puzzle, but some pieces are missing.
Makes sense. So how does variational causal inference help?
It combines 2 key sources of information.
Okay.
First, the individual's observed outcome, the actual data we have.
Learning from what actually happened.
Exactly. And then the response distribution of similar individuals who were exposed to the treatment or condition we're interested in.
And we're looking at a group of comparable individuals to see how they reacted.
Right. By combining this individual and group level information, it can make more accurate predictions even with limited covariates.
So it's like filling in those missing puzzle pieces by looking at how others solved similar puzzles.
The second paper, Predicting Cellular Responses with Variational Causal Inference and Refined Relational Information, uses this concept to solve a very specific problem, predicting a cell's gene expression under counterfactual perturbations,
Counterfactual meaning.
conditions the cell hasn't actually experienced.
So like predicting how a cell would respond to a drug without actually giving it the drug.
Exactly. We can explore what-if scenarios without having to do the actual experiment.
Wow, and how does this work?
It incorporates something called gene regulatory networks, GRNs,
Okay.
which map out all the relationships between genes, showing how they regulate each other.
So it's like a roadmap of how genes interact and control each other.
Think of a city's transportation system.
Okay.
Genes are like destinations, and regulatory elements are like the traffic signals controlling the flow of traffic.
Cool analogy. So how do these GRNs help make better predictions?
They provide essential biological context. By understanding how genes interact, the model can make more informed predictions about gene expression under different conditions.
Like having a guide to navigate the complex world of gene regulation?
And here's the cool part. The researchers actually refined the GRNs during the model's training.
So the model is learning and updating its understanding of how genes interact.
Exactly. This led to more accurate predictions and even new insights into gene relationships.
Impressive. So the model's not just predicting, but also helping us understand biology better.
They also developed a robust estimator for something called marginal perturbation effect.
Marginal perturbation effect.
It basically measures the overall impact of a perturbation on gene expression.
So not just whether a gene's expression increases or decreases, but by how much.
Precisely. It gives us a much more complete picture of how perturbations affect the cell.
Really impressive. These two papers highlight the amazing potential of computational approaches.
Right. They're helping us unravel complex biological processes and make predictions that were once impossible.
It feels like we're entering a new era of biology where computational tools are going to be crucial.
It's all thanks to the incredible complexity of the genome.
There's so much more to explore and understand.
And that wraps up today's Genomics Daily Brief.
Wow, we covered a lot today.
From unexpected origins of satellite DNA to the power of variational causal inference.
A lot of exciting research happening in genomics.
Make sure to check out the show notes for links to all the papers we discussed.
Definitely worth a read.
And join us tomorrow for another deep dive into the cutting edge of genomics research.
Until then, keep exploring the fascinating world of genomics.